Okay, out of the cell surface receptors, the first one that I'm going to explain is GPCR. And as I said, the GPCRs are the oldest class of cell surface receptors and they're the most common. So in many cases, in many of the tissues that you're going to check, you're going to find the GPCRs. And of course, if you're going to do drug design, if you're going to do drug repurposing or drug delivery, you have to know the GPCRs really good because 1/3 of all the drugs that we have, they are actually targeting GPCR mechanisms. So it is that important. So you better understand this GPCRs real good. So in our body, in the human body, there are more than 700 GPCRs. And believe me, this is not a high number because we know that the dogs have more than 900 just for the smell. So in just for their nose, they have more than 900 GPCRs to understand the smell, to understand what they're smelling. And we always think that the dogs have the best smell, but no, rats actually are the mammals that have one of the best smells. They have more than 1000 GPCRs in their noses, so they can discriminate a very large class of odors. But we cannot use rats for the detection of narcotics, mostly because we cannot educate the rats. So although they can understand there's narcotic, there is something strange going on, they cannot signal us. Oh, the GPCRs are also very useful because, well, first, the GPCRs are 7 transmembrane proteins. So 1, 2, 3, 4, 5, 6, 7. All members of this GPCRs, they have this 7 transmembrane structure. This is also why we call them 7TM proteins. And you can imagine their primary structure: ER start, ER stop, ER start, ER stop, ER start, ER stop, goes on. And of course, they consist of this hydrophobic and hydrophilic residues. The residues that are facing this here, the membrane, they are hydrophobic, and inside they are hydrophilic. So it's a combination of hydrophilic and hydrophobic amino acids. And what is nice is that the top of the GPCR, this portion creates a pocket, and this pocket receives the signal. Because this pocket is large and it can have any kind of structure, it can have any kind of shape, it can recognize any kind of signal. So they are not that rich with the enzyme coupled receptors or ligand coupled ion channels, ligand gated ion channels, but for the GPCRs, they can really respond to anything. They can respond to neurotransmitters. So we have GPCRs on the neurons. They can react to proteins, real large proteins. They can get the mediators, hormones. They can get small peptides. They can even recognize the fatty acids or the small hydrophobic molecules. So they can easily recognize anything. This is why we don't call them family. We call them superfamily because they are so various. And the only common thing that between them is that they all have the 7 transmembrane domain, and they all function as receptors, the cell surface receptors. But except that, there are not many things common. Oh, I forgot something. They can even recognize light. Great. So unlike chemicals, proteins, hormones, they can even recognize lights. This is how our eyes are seeing. Our eyes have light inactivated GPCRs. This is how we get the light. And maybe you remember the very basis of I hope you remember mitochondria, how the energy is produced in mitochondria. They are creating pumps. This hydrogen is pumped between the matrix here to the intermembrane space. And then when the protons are coming back, it creates energy. So this actually exists in some archaea, and they have this light-activated proton pump. That proton pump is also GPCR. So that archaeobacteria has bacteriorhodopsin. It's a light-driven proton pump, proton pump, and it does not work through G protein. There is no G protein. It's a simple pump. It also shows that the GPCRs evolved much before than G proteins themselves. Okay, the basic working mechanisms. The mechanism. First, G protein, as the name implies, interacts with the GTP. And here, this is not a monomeric GTPase. This is a trimeric G protein. Trimeric G protein means it has three parts: alpha, beta, and gamma. Alpha and gamma, they are membrane tethered, which means post-translationally in the Golgi, some lipid groups are added to these proteins. This is why, although they are not transmembrane proteins, although no part of this amino acids are passing through membrane, they are staying on the membrane because of these little lipid groups. Okay. And the second thing is that in the inactive form, there is the GDP. And GDP has two phosphates. It's low in energy. In the active form, it doesn't have GDP. It has GTP, which means, remember the activation and inactivation of the GTPases, which means a GAF acted on them. In this case, the GAF is the G protein. So it pushes the GDP away, it removes the GDP away. So when the GDP is removed, a GTP from the surrounding joins, because it is the intracellular cell, and in the cell inside there are lots and lots of GTP available everywhere. So first, this is the trimeric G protein. Second, it is activated by the GPCR. Third, when it is activated, a GTP joins. And fourth, when it is activated, it doesn't stay trimeric anymore. It is going to be divided into two. So, in one part, there is the alpha subunit, and the other part, there is the beta and gamma subunit. So, beta and gamma stay together. This is why we call them beta gamma. So, they are going to separate into two. So as long as it has GTP, they are going to stay separate. Once this GTP is hydrolyzed and it becomes GDP, they are going to come back together. So let's see how it affects how it is affected by the signal coming from outside. So when there is no signal coming from outside, the GPCR is inactive. No signal. When the signal joins, the GPCR becomes active. Of course, it's not only going to stay active, but it's going to relay the signal to the intracellular site. There's a transmembrane protein, there's a cell surface receptor. It has to communicate inside the cell. So inside the cell, it is going to give this information to the G protein. And how it gives this information? It just kicks out the GDP. When it kicks out the GDP, a GTP joins, and then the G protein is broken into two, and voila, now the signal is inside the cell, because now we have two active compartments. This one is active, so this is going to interact with many other molecules inside the cell, and it's going to communicate many things. But these two are also active, so they are both active when they are separate, and they are inactive when they are together. So it's like a boring couple. When they are together, they are really boring. Then they don't do anything. But when you separate them into two individually, each of them is very funny. Okay, so they are going to continue working until this GTP is hydrolyzed. Now, hydrolyzing this GTP is actually quite fast. It just takes seconds. But within the seconds, in the molecular term, second is not something short actually. Many things happen. It's going to create lots of reactions. It's going to activate several things, and then it is going to be inactivated. If you cannot inactivate it, if it stays this way, it's a problem. This is actually what some microbes, some pathogens are doing. They block this G protein from coming back. They block the inactivation of this G protein, and at the end, people die either from diarrhea or from coughing or something, but it's little. I don't know why they come back. Okay, yeah, here you can see their activity. Yeah, here they become inactive again. This GTP is used up, the phosphate group leaves. Once the phosphate group leaves, it stays with GDP like a broke man who spent all its money, so he turns back to his wife. So they are all together family again, and they are inactive again. Time to see, well, I said that there are some GPCRs on the surfaces, on the postsynaptic surfaces. Yes, they also recognize the neurotransmitters. Yes, they can also act on the synapses, but it is indirect. So think about this ligand-gated neurotransmitter channels or ligand-gated channels. They are quite direct. As soon as they interact with the neurotransmitter, it's going to open the channel and it's going to change the membrane potential. But here in this case, a series of events has to take place in order for the membrane to open. So first, the signal is going to activate the GPCR. The GPCR is going to activate the G protein. The beta gamma complex is going to activate the ion channel, and then it's going to change the membrane potential. And here it also acts on membrane potential, and not only that, here there are some other activities going on. So in a way, having these GPCRs on the neurons have double purposes. It both creates the membrane potential, plus it creates some long-term effect, which is going to change the profile of the proteins inside. Okay. Yeah. So let's see what other things can happen. What is the downstream pathway? And when we say upstream, downstream, it is not like upstream or downstream of the DNA anymore. Of course, in the DNA, it's all about where the promoter is. It's all about the direction of the transcription. But here, upstream means the signal. So when you have the signal, it is upstream. And when it is relayed into the cell, and there are many effectors, so it is protein to protein, effector to effector, this is called downstream of the pathway. Okay. The upstream of the GPCR is more or less standard, so they get the signal, they activate the G protein, but downstream of this pathway is very complex. First of all, when this alpha subunit is free, when the alpha subunit is active, what it is going to do, that it's going to activate an enzyme. It can be a guanylyl cyclase or adenylyl cyclase, and this enzyme is going to create second messengers. Now, please don't call it secondary messengers. It's not called secondary messengers. It is second messenger. And the second messengers are very useful. First, because they are small, see their size in compared to these large things. They are not membrane attached, they are not large. They are really small, they can diffuse easily. So once you have a second messenger here, the number is quite high in compared to the previous signals. Like many of them is here. And they are going to be distributed everywhere in the cell. So the same signal is distributed all around the cell. And third, they are short-lived. So in 5 seconds after having these second messengers, the second messenger is going to be inactivated. And once it is inactivated, the signal is off. So why to have the second messengers? To amplify the signal. So instead of, there is one single molecule received and thousands of second messengers produced. Second, it is received from one single place here, but it is distributed everywhere. And third, it's a wave of signal. It is just going to be activated and then inactivated really fast. So the common second messengers that we have, the famous ones are cyclic AMP, cyclic GMP, as we have seen them with the Viagra and in the heart case, and calcium. Of course, there are many more, but these are the most famous ones. Oh, let's first see a very common one, cyclic AMP. This is the ATP molecule, the famous ATP molecule that you see everywhere. Now, when the adenylyl cyclase enzyme interacts with that, by the way, this is the enzyme. This is a membrane tethered enzyme which is directly activated by the alpha subunit of the G protein. So, when the alpha subunit of the G protein activates adenylyl cyclase, it is going to create cyclic AMP. Now, this is the second messenger. And this second messenger is very active, very small. But as I said, it is also short-lived because soon it is going to be inactivated by cyclic AMP phosphodiesterase. The second messenger is going to survive for a few seconds, and then it is going to be inactivated by cyclic AMP phosphodiesterase. Now, what coffee does is that it stops this enzyme. This enzyme is not active anymore when you drink coffee. This is why the second messenger cyclic AMP is active for longer. Instead of 2 seconds, let's say it is active for 10 seconds. And it is going to create a longer signal. This is why you feel more energetic. Of course, on different tissues, it have different effects. Of course, the effect in the brain in different places can be different. What I'm telling is a very fundamental, very basic way how coffee works. It just blocks the cyclic AMP phosphodiesterase, which is the enzyme killing cyclic AMP. So you kill the killer, so the cyclic AMP survives more. Now we are going to continue more, but I have to finish